What I wish I knew at 16. The human brain isn't just weird, it's a timeline of epic discoveries. Here's how we cracked the code, one neuron at a time. It all started in ancient Egypt, where people thought the heart did the thinking. Fast forward to the 1600s, Descartes guessed the brain was a pump for animal spirits. But in the 1800s, things got real. Scientists like Golgi and Kajal peered through microscopes and discovered brain cells, neurons. Suddenly we knew thoughts traveled as tiny electrical zaps. By the 20th century, we figured out how neurons talk to each other using chemicals called neurotransmitters. Now, with high-powered scanners, we can watch memories form in real time. Oh, crazy, right? So next time your brain's overloaded before a big test, just remember, your neurons are working overtime and it took centuries to uncover.
2. Structure of brain cells. A. Neurons, nerve cells. Neurons are specialized for transmitting electrochemical signals. While there are variations in their exact shape and branching patterns, a typical neuron has three main parts: soma, cell body. This is the central part of the neuron, containing the nucleus and other essential organelles like mitochondria and the Golgi apparatus, responsible for maintaining cell function and protein synthesis. Dendrites. These are tree-like branching extensions that protrude from the soma. Their primary function is to receive incoming electrical and chemical signals from other neurons. They are covered in synapses, which are the points of communication. Axon. This is a long tail-like projection that extends from the soma at a specialized region called the axon hillock. The axon's main role is to transmit electrical impulses, action potentials, away from the cell body to other neurons, muscles, or glands. Myelin sheath. Many axons are covered by a fatty insulating layer called the myelin sheath, which is formed by glial cells, oligodendrocytes in the CNS and Schwann cells in the PNS. This sheath acts like insulation around an electrical wire, greatly increasing the speed at which nerve impulses travel down the axon. Nodes of Ranvier. These are small gaps in the myelin sheath. Electrical signals jump between these nodes, further speeding up transmission. This process is called saltatory conduction. Axon terminals, terminal buttons. Located at the end of the axon, these specialized structures contain synaptic vesicles that store neurotransmitters. When an action potential reaches the axon terminal, neurotransmitters are released into the synapse to communicate with the next cell. Synapse. This is the tiny gap between the axon terminal of one neuron and the dendrite or soma of another neuron, or a muscle, gland cell. It's the site where chemical communication via neurotransmitters occurs.
B. Glial cells. Neuroglia. Glial cells are non-neuronal cells that provide crucial support and protection for neurons. They do not transmit electrical signals in the same way as neurons, but they actively participate in brain signaling and are essential for neuronal health and efficiency. IRA. Types and functions of brain cells. A. Neurons. Neurons are broadly classified based on their function and structure. By function. Sensory neurons, efferent neurons. These neurons transmit signals from sensory receptors, for example in the skin, eyes, ears, to the central nervous system, brain and spinal cord. They allow us to perceive and interpret our environment, for example touch, pain, light, sound. Motor neurons, efferent neurons. Efferent neurons. These neurons carry impulses from the central nervous system to muscles and glands, controlling movement and bodily functions. They can be subdivided into upper motor neurons, brain to spinal cord, and lower motor neurons, spinal cord to muscles. Inner neurons, relay neurons. These neurons act as intermediaries, connecting sensory and motor neurons within the central nervous system. They are crucial for processing information, forming neural circuits, and enabling complex thought, learning, and memory. They can also communicate with each other to form intricate circuits. By structure, multipolar neurons. The most common type, characterized by one axon and multiple dendrites extending from the cell body. Most motor neurons and interneurons are multipolar. Bipolar neurons have one axon and one dendrite extending from opposite ends of the cell body, found in specialized sensory organs like the retina and olfactory epithelium. Unipolar neurons have a single process extending from the cell body that then branches into an axon and a dendrite. Exclusively sensory neurons, typically found in ganglia outside the central nervous system. Pseudonipolar neurons appear to have one process, but it's actually two fused processes that act as both an axon and a dendrite. Most sensory neurons are pseudopolar, and axonic neurons lack a distinct axon, and all processes appear as dendrites. Their function is not fully understood, but they are thought to regulate electrical activity within the brain.